content.
Yeah, there are some questions.
Oh, we have somebody with.
Okay, good afternoon, everyone. Thanks for coming. It's a busy time of the year, and today it's my pleasure to introduce Tim Herr, Michigan University. There's a big difference, right? University of Michigan. Studied to give a background in CAU, Chinese Agricultural University, China Agricultural University, I think the major agricultural university in China, and he did his master's in Chinese Academy of Sciences. And I realized today he has 2 PhDs. That's why I know he's smart. One,
You only get a PhD chips.
no. One in CSU, right?
When is CS?
Okay, Chinese Academy of Sciences in plant pathology or genetics.
And genetics.
Genetics, and then he came over to the US in Kansas State University, where famous people like Ron gave PhD.
Famous team.
He's also PhD from Kansas State, right? The same as that place. Then he went to Harvard to do a postdoc, Harvard Medical School. And then I don't know the dates, but he went to be a faculty, Texas, and then the Department of Biophysics and
Biochemistry, yes.
And recently he moved to Michigan and joined the Department of Molecular, Cellular, and Developmental Biology. I assume this is a general biology, not the plant biology.
So that's right.
Yeah. And a lot of you probably know, Tim publishes extensively in very, very high impact papers, there's possible Science Nature. He's funded by NIH and NSF. And today, I'm thankful that he drove here to share his exciting results. With that, I leave the floor to Tim.
Thank you. Very nice introduction. It's so good to be close here and I can drive here. And so far, really fantastic time to meet older friends and know new friends. Yeah. So my lab mainly work on plant immune signaling. So in particular, we designed different genetics and in model plant alloptopsis and try to understand how the effective immune response is activated, and also how the activated immune response is maintained. Like a plant, like other organisms, need to have like effective defense to survive. However, like overactivation of defense also could be a problem, detrimental to the hosts and cause like death. Yeah, we call it autoimmunity. So this defense and over defense need to be like maintained at a certain level. Okay, so this we call it reconciliation of defense and defense is controlled by gene expression and transcriptional, post-transcriptional, translational, and post-translational level. In addition, my lab also work on some like functional and translational genomics of cotton stress response, especially like the interaction between cotton and physalis of sporin, and also improve like cotton like a stress response to biotic and abiotic stress through like translational genomics. So I would like to start with this general introduction or background to offer some like a historical overview of our understanding of plant immunity. So the concept of plant pathology emerged in the middle of 19th century, which was about a decade earlier than the germ theory for animal disease. Then building upon the understanding of genetics and inheritance, in 1940s, Bloch proposed this famous gene resistance model to explain the plant resistance is governed by a resistance gene in plant and also a ABR gene in pathogens. So this interaction called ABR-R interaction is highly specific occurs at a specific plant cultivar level and also specific to pathogen races. So we call it race-specific resistance. And 1960 or 1970 marked the discovery of a group of pathogen components, which were named elicitors in 1984. So these elicitors could trigger general immune or defense response attack, such as production of phytoalexin in plants. And this response occurs at the plant species level and also not specific to certain pathogen like races or cultivars. So we call it a non-specific, non-race specific plant defense. And 1984 was also a year like first the AVR gene, AVR-A was cloned. And then in 1990s, like there was a blooming of the cloning of plant-resistant genes, R genes. Actually, this is a PTO cloned by Greg Martin right before he moved here as a faculty member. But most like R genes are nucleotide binding site, NBS, AIR proteins that could interact with bacteria AVR genes directly or indirectly. And then in the early like 2000, Soster and colleagues like revealed a striking similarity of the molecular mechanism of animal and plant innate immunity, and proposed to use this term, which was used in animal immunity, to refer this PAM, pathogen-associated molecular patterns, for these general elicitors. Okay, this also explained why like plant could respond to this PAM or general elicitors at the species level. And meanwhile, the several PRRs, pattern recognition receptors, which could recognize these general elicitors or PAMs, were cloned at 2000. The first cloned PR was FS2, which recognized bacteria flagellum. And one of the most exciting like discovery recently in plant immunity is the discovery of plant like NAR resistant cell. So upon activation, plant NAR already rise and form this resistant cell, which is essential for the activation of defense. Okay, with this introduction, we know plant immunity is kind of like a two-branched system. One branch is mediated by this cell surface resident pattern recognition receptors, which are usually receptor kinase, it's also called receptor-like kinase, IK or ILK, or receptor-like proteins, recognize conserved pathogen or microbe associated molecular patterns and trigger this so-called pattern triggered immunity, PTI. Another group of plant immunity is mediated by this intracellular nucleotide binding site using rich repeated protein, NBS, ARR, or not like a protein, or classically like resistant proteins. And they recognize pathogen encoder effectors and initiate this so-called effector triggered immunity, also classically like a gene for gene resistance. The recent study also suggests the interaction or mutual potentiation between these two branches of immunity to build a robust plant immune response. These two groups of plant immune receptors also functionally and also structurally similar to animal like immune receptors, with receptor-like kinase or receptor-like proteins similar with toll-like receptors that perceive extracellular MEMS, and NBS AR proteins similar with not-like receptor protein perceived like intracellular PAM and involved in the inflammation and also program cell death. There are also certain similarity of downstream signaling events, such as activation of MAP kinase cascade and transcription factors or production of antibacterial compounds, and also induction of program cell death in plant and animal like innate immunity. However, we think that this similarity is due to the convergent, not conserve the evolution in plant and animal innate immunity. So when I started my lab at the Texas A&M University, we designed a highly sensitive and quantitative genetic screen to understand immune gene regulation. So in this screen, we fuse the promoter of a gene called FRK1, which is quickly induced by multiple elicitors such as bacteria flagella. And then we fuse this gene promoter with luciferase reporter and then transform it into Arabidopsis mutants, and then looking for transforming into Arabidopsis plants and then perform its genetics, then looking for the mutants with either reduced or enhanced luciferase activity. We call these genes Arabidopsis genes governing immune gene expression, ARGEs. So you all know Texas A&M students are ARGEs. So this is a good project to educate and attract many ARGE graduate and undergraduate students. And also nowadays, this NGS also rejuvenate genetic screen for the rapid gene cloning. And after we identify gene, we will use our well-established CR-based system for in-depth biochemical and cellular characterization of gene functions. And then we use intact plant-pest interaction at the organism level to confirm what we find in the chloroplast. So this group of screen quite successful. We identified many genes and reviewed layered transcriptional, post-transcriptional, translational, and post-translational regulation in plant immunity. So I'm not going to go details about our ARGE screens, and if you're interested, we have some publications about this part. And I talked about the PRs are receptors for pathogen elicitors. They are very specific to recognize individual elicitors. However, there's a group of receptor kinase called SERK, somatic amelogenesis receptor kinase. This group of receptor kinase could function as co-receptors that interact with multiple PRRs or receptor kinase involved in plant immunity. For example, here, SERK3, also called BAC1, together with SERK4, could complex with PRRs that perceive cells from bacteria or mycete, fungi, and even plant-derived peptides. Okay, so this functions as a co-receptor of multiple PRRs. In addition to these PRRs, this BAC and SERK family receptor kinase also function as a co-receptor, interact with receptors involved in plant growth and development, stomata patterning, organ abscission, and also hormone signaling. Actually, BAC1 was originally identified as the BRY1 co-receptor, which BRY1 is a receptor of hormone plus in a steroid. So this SERK and BAC families are shared co-receptor in plant immunity and development. They are important immune regulators. However, if we delete BAC1 and SERK4, they also cause lethality. So this lethality is due to the overactivation of defense. As you can see, this carbon blue staining, see the production of this precipitate, the overactivation of defense. So here, many things, too much defense is also as bad as too little. So this defense needs to be tightly controlled. So this BAC1 SERK4 could function as a co-receptor of multiple PRs. They are also important in maintaining immune homeostasis. So to understand how this BAC1 SERK4 involved in maintaining homeostasis, we developed an innovative genetic screen for suppressors of BAC1 SERK4 cell death. So in this screen, we silenced BAC1 and SERK4 with virus-induced gene silencing, which is a type of IAI. So this silenced the plants phenotypically similar with genetic mutants, although not as strong as genetic mutants, but at least show cell death. And then we screen Arabidopsis TDNA library with this VRGS, and then looking for the mutant which could suppress BAC1 SIK4 cell death. So we call this mutant back to life, BTL. For example, here, the first mutant we screen is this STT3A, which is a catalytic subunit of oligosaccharidotransferase complex, okay, involved in the protein N-glycylation. As you can see here, this STT3A mutant suppressed BAC1 INAI BAC1 SIK4 mediated cell death. This protein glycylation is connected with ER quality control, and both N-glycylation and ER quality control are mediated by a series of enzymatic reactions. And then with VRGS towards the mutants involved in this process, we find that some specific glycan modification and also components involved in the ER quality control are involved in the BAC1 SIK4, like mediator cell death. So another mutant that we call the BTL1 include a cyclic nucleotide gated channel, CNGC20, which we show that it is a permeable casein channel. And here you can see that the three CNGC20 mutants suppressed INAI BAC1 SIK4 induced cell death. So in this paper, we show that this BAC1 SIK4 as a shared receptor and associate and transphosphorate with multiple receptors, and they all receptor kinases involved in immunity and growth. However, this BAC1 SIK4 could also phosphorate CNGC920 and its close homolog 19, and this phosphorylation lead to the degradation of these two CNGC proteins, which keep casein at a normal level. However, when BAC1 and SIK4 are absent, then this CNGC19 and 20 overproduced, which lead to the massive influx of casein and downstream signaling, and could cause the cell death. This BTL2 mutant, I spent a little bit more time about it, and you can see here 3 alleles of BTL mutant suppressed VRGS BAC1 SIK4 induced cell death, and genetically BTL2 mutant also suppressed BAC1 SIK4 double mutant mediated cell death. As you can see here, the triple mutant grew much better than double mutant, and the cell death much reduced, and also overproduction of H2O2 is much reduced in the triple mutant compared to the double mutant. And this triple mutant could normally grow, develop a stem, and set up seeds. Actually, this is the best suppressor we identified so far. So we find that this BTL2 gene is highly induced in the BAC1 SIK4 mutant. Then we asked whether this overproduction of BTL2 is connected with BAC1 and SIK4 cell death. So here we overexpression always on the native promoter, but still overexpress the BTL2 in transgenic plants. And we saw plants show different level of cell death, with 40% of plants show different level of necrosis and also the cell death. And also this severity of the cell death is correlated with the protein expression level. So here we know BTL2 overexpression could induce cell death, and BTL2 is a cell death inducer. BTL2 is an uncharacterized receptor kinase with an extracellular leucine-rich repeated domain, LRRK, which is similar with BAC1 SIK4 and also some other PRRs such as FS2, perceived bacterial flagellum. And like many IAKs, BTL2 also localized on the plasma membrane. And also we show that BTL2 is the active kinase with this in vitro kinase assay. The kinase mutant lost the kinase activity. When we transfer the kinase mutant into the BTL2 transport complementation, and we could not see the cell death upon silencing BAC1 and SIK4. This indicated that BTR2 kinase activity is essential in mediating like a BAC1/4 induced cell death. BTR2 is a kinase. Usually kinase has the autophosphorylation activity. We identified T669 as the autophosphorylation site. As you can see here, the mutation of this site to alanine block its autophosphorylation activity. And then when we transfer this BTR2 T69 mutant into BAC1/4 and the BTR2 mutant for complementation. Again, this mutant could not complement the lethality as Y-type BTR2. So this is like a Blues defect, and also this is the cell death. The autophosphorylation mutant could not complement the cell death and overproduction of H2O2 compared to the Y-type BTR2. So this indicated that the BTR2 autophosphorylation is important for its ability to trigger the cell death or autoimmunity. As I mentioned, BTR2 is an IRK BAC1 associated with many IRKs as a co-receptor. So here we asked whether BAC1 could also associate with BTR2 with this like a co-IP asset in like transgenic plants with like a native promoter of BTR2 and also BAC1 antibody. We show that BTR2 could co-precipitate with BAC1, and also in vitro BTR2 and the BAC1 kinase domain they interact directly, and the kinase activity seems like is important for mediate these two receptor kinase and interaction. We also identified this S673 is a phosphorylation site of BTI2 by BAC1. Okay, previous site was BTI2 autophosphorylation site, which is essential for the cell death. So this is a BAC1 phosphorylation site, and we transferred this phosphomimic form, which changed to like S676 to D, and this phosphomimic form into the BAC1/4 BTI2 mutant. And this phosphomimic form could not complement this cell death inducibility compared to the Y-type BTI2. So this indicated that BAC1 phosphorylate BTI2 and then suppress this cell death inducibility. This also explains why this BTI2 does not induce cell death in Y-type, because BAC1 phosphorylate BTI2 and the BTI2 cell death inducibility is suppressed. And just now I showed that CNGC is important in triggering cell death. However, we find that when we overexpressing CNGC20 is not sufficient to cause the cell death, then CNGC activity must be regulated by other components. So here we find that when we co-express BTI2 and the CNGC in Nicotiana Pennsylvania, these two proteins cause strong cell death, and also this cell death is suppressed by the BAC1. And we find that overexpressing BTI2 cause the cell death depends on CNGC. So you can see here previously 40% of BTI2 transgenic plants cause cell death in Y-type. Here only about 5% of plants show certain growth defect, no always cell death. So this indicates that BTI2 mediated cell death depends on the CNGC. We also show that BTI2 could interact with CNGC20, and also BTI2 could phosphorylate CNGC20. And more importantly, when we co-express BTI2 with CNGC and not BTI2 kinase mutant, this BTI2 could enhance the CNGC20 channel activity with this like a patch clamp recorded in Xenopus outside. So this indicated that BTI2 could phosphorylate CNGC and then activate channel activity and then induce the cell death through passive like a calcium influx. Then what's the physiological function of BTI2 in Y-type plants? We try to determine whether they have any defect in like PTI, ETI, but we did not see like any obvious phenotype associated with the immunity for the BTI2 single mutant. And one report previously by another lab showed that like BAC1 has enhanced the immune response, as I show you here with this signaling blues inhibition in response to this peptide we call the phytocytokine type 1. Okay, so phytocytokine is type of damage associated molecular pattern, but they are mainly derived from plant cells. Okay, it's a small secret protein, and it has immunomodulating functions, and also similar with animal cytokine. So that's why we call it the phytocytokine. Basically, it's plant-derived small peptide, and we call this immune response triggered by dam of phytocytokine as danger-triggered immunity, DTI. Okay, so this type 1 is a phytocytokine. It is like perceived by the receptor type R1 and R2, which is also a ARR receptor kinase, and also use BAC1 as a co-receptor. So here it is very puzzling. As a co-receptor, why the depletion of BAC1 triggers stronger this type of immune response? So we find that this immune response is mediated actually by BTL2. As you can see here, in the BAC1 and the BTL2 double mutant, the type 1 triggered stronger immune response is kind of like completely blocked. And also this BAC1 BTL2 double mutant show the compromised response to the type 1 treatment. And also the BTL2 BAC1 CERC4 triple mutant even further like reduce the response to the type 1 treatment. And BTL2 single mutant itself does not have any like phenotype. Similarly, use this MAP kinase asset, we show that the type 1 triggered MAP kinase activation is reduced in the BAC1 BTL2 double mutant, and it was further reduced in the BAC1 CERC4 BTL2 and triple mutant. And again, we show that this BTL2 triggered like a cell death depends on the type R and the type 2 receptor. When we overexpress this BTL2 in the type mutant, the cell death was much reduced compared to the wild type plants. So here, the information we got is that in the absence of the BAC1, BTL2 trigger this PEP response, and this PEP response also depends on the receptor type R1. Okay. So this BAC1 CERC4 mutants, there are many like phytocytokine genes are induced, and in addition to like PEPs, another group of phytocytokines called SCOOP were also highly induced in the BAC1 and the CERC4 mutant. And we tested whether this BTL2 is also involved in phytocytokine signaling. BTL2 single mutant does not have any defect in phytocytokine like triggered MAP kinase, RS production, or callus deposition. However, BAC1 BTL2 double mutant show very severe defect in SCOOP-induced MAP kinase, RS, and callus deposition. So here, again, the data suggests that when BAC1 is depleted and the BTL2 could function substitute like a BAC1 and trigger this immune response. This immune response was blocked double mutant. And I forgot to mention that SCOOP also use this receptor-like kinase MYC2 as a co-receptor, and also use MYC2 as the receptor, and use BAC1 as a co-receptor. So SCOOP could also induce MYC2 and BAC1 interaction. Since BAC1 is the co-receptor of MYC2 and type 1, we here asked whether BTL2 could interact with MYC2 and type R1. Use this Fred Frame assay, we can see that BTL2 is in close proximity with MYC2 and PAPA1, two phytocytokine receptors. And then use this CoIP assay, we show that BTR2 associated with MYC2, and also this association was enhanced upon like spoof treatment. So this BTR2 kind of like a similar with a BAC1 in terms of its function. So here, what we show that BTR2 associate with multiple phytocytokine receptors, at least MYC2 and PAPA1, and also potentiate their signaling in the immune-compromised BAC1 mutant. And the BTR2 may function as an alternative co-receptor of phytocytokine receptors when BAC1 SIK4 are absent. Consistent with this hypothesis, we find that the triple mutant BAC1 SIK4 BTR2, when all these three co-receptors absent, the immune response was further like compromised compared to BAC1 SIK4 and the double mutant. So here we identify the BTR2 as an important regulate in BAC1 SIK4 mediated like cell death. Then what's the physiological function? So I summarize here, like in a wide type of plants, like BAC1 SIK4 are key like immune co-receptors associated with multiple PRRs and trigger plant PTI. However, this BAC1 SIK4 could also phosphorate this S673 and suppress this BTR2 activity and keep it inactive. And at the same time, it also could phosphorate CNGC19 and 20 and keep the protein abundance low. However, since this BAC1 SIK4 are important co-receptors, a lot of pathogens could use effectors to target a BAC1 and SIK4 to degrade it or to duplicate it and whatever to destroy it. And then when this happens, and this BTR2 suppression is released, okay, the BTR2 gets activated. This activation mainly induces the T669 autophosphorylation. Upon this autophosphorylated, and it could activate the calcium channel CNGC, induce calcium influx, and also induce the production of phytocytokines. So these phytocytokines could be perceived by phytocytokine receptors, and this phytocytokine receptor could interact with BTI2 and activate, we call it like DTI. This amplification of DTI and also massive influx of calcium could lead to like autoimmunity. So this BTI2 is important in balance like immunity and autoimmunity through a phosphor switch by BAC1. So this is one type of screen use BAC1 SIK4 as readout, and another type of screen we use this like MAP kinase cascade. So MAP kinase cascades are important like signaling modules in plant immunity. So we know like perception of the PAM or MAMs activate two MAP kinase cascades, include activate the MPK3, 6, and also MPK4, these two cascades. And interestingly, deletion of this MAP kinase 4 cascade, including MPK1, MKK2, and MPK4, also cause lethality. This lethality again is caused by the activation of defense. And we know this defense is mediated by NAR protein, SUMO2, and also by MAP triple kinase, MEKK2. So here we are interested in how PRR activated MAP kinase could suppress NAR SUMO2 mediated immunity. This could help us to understand the interconnected PR and NAR interaction or activation in maintaining like immune homeostasis. To address this, we use the same approach, INR genetic screen, to looking for the MPKK1 suppressors. As you can see here, again, VIGS MPKK1 could also cause cell death. Phenol copies like MPKK1 genetic mutant. Okay, and then we screen the anaptopsis tDNA library to looking for the suppressors of MEKK1 CRDs. And we call this mutant lethality suppressor of MEKK1. As you can see here, the LAT1 mutant suppressed the RNAi MEKK1 induced growth defect and CRDs and the overproduction of H2O2 and also expression of PR1 gene. And also this suppression was confirmed in the LAT1 and MEKK1 genetic double mutant. So what is LAT1? LAT1 includes a mylectin-like receptor kinase with two actual mylectin-like domains, which is different with FACT1 or other FS2, this AR ILKs. Actually, they belong to the same group of Fronia and Anza, and they have team members, which Shana is working for the Fronia. And this group of genes are named by different mythologies, like Fronia is goddess of fertility. They are our gene name, Latin also is the personification of death in Rome mythology. Okay. Then towards the targeted screen of this group of receptor kinase, we find another mylectin-like receptor kinase we call the LAT2, also involved in MEKK1 mediated cell death. We further show that LAT1 and LAT2 could complex with each other on the plasma membrane, and they form this kind of a receptor complex in mediating NAR SUMO2 activation. So here we also provide genetic evidence, direct evidence to show the connection between receptor kinase and also the NAR protein activation. So interestingly, we find that actually LAT2 could promote LAT1 phosphorylation, and show you here with this geomobility shift in this immunoprotein, and also the GPR anchored protein ALG could directly interact with the extracellular domain of LAT1 and LAT2, and this in the ALG mutant, the plasma membrane localization of LAT1 and LAT-T seem likely greatly impact. A lot of this LAT1 and LAT2 GFP are stuck in the ER. So here, most likely, this ALG could function as a chaperone or co-receptor of this LAT1 and LAT2 and assist these receptors delivered to the plasma membrane, and where it could activate the downstream NAR and SUMO2. Then how SUMO2 is activated? Then we find that this triple kinase MEKK2, although it was annotated as a kinase, but it does not have the kinase activity, and it functions as a pseudo kinase, stabilize and scaffold both this ILK, LAT1, and also the NAR SUMO2. SUMO2 could be ubiquitinated by the F-box protein CPR1, and show you here this SUMO2 ubiquitination is reduced in the CPR1 mutant. However, this SUMO2 could counteract with CPR1 mediated ubiquitination and then stabilize it. As you can see here, this ubiquitination is enhanced in the MEKK2 mutant, suggesting that the Y-type MEKK1 could suppress this ubiquitination. So here we show that this stability of this NAR protein SUMO2 is oppositely regulated by CPR1, which promotes degradation and keep a plant alive, and by this pseudokinase MEKK2 for stabilization and also for activation and cause autoimmunity. Another mutant that we identified called LAT3, which include plant U-box E3 ligase TAB. As you can see here, this LAT3 again suppress the MEKK1 cell death by VIGS. Also genetically, LAT1 or LAT3 suppress the MEKK1, MKK12, MPK4 cell death in double and triple mutants, suggesting that this LAT3 function downstream of this MAP kinase cascade. To understand this how LAT3 function in this pathway, we performed the proteomics, they identified this CRCK3, which is a chemodulary binding receptor-like cytoplasmic kinase, and interact with LAT3. So as you can see here, they are in close proximity with this Brad stream assay, and also they could interact directly with in vitro put-on assay, and this interaction occurs through this arm domain of this LAT3. An interesting phenomenon we observed is that when we overexpression of CRCK3 in a LAT3 mutant, this excessive CRCK3 could complement LAT3 defect in triggering cell death. So this indicated that the defect of LAT3 is due to the reduced expression of CRCK3. So indeed, we find that the expression level of CRCK3 is much reduced in the LAT3 mutant with this a GFP image, also Western blot. And also, when we co-express of LAT3 with CRCK3, the protein level of CRCK3 is increased. So this indicated that LAT3 could stabilize CRCK3. And this is quite puzzling. We know E3 ligase usually degraded protein substrate, yeah. Then how E3 ligase could stabilize the protein? So we try to determine whether LAT3 is active E3 ligase. With many attempts, we could not detect E3 ligase activity. This PUB13 is a positive control. And also we mutated conserved E2 binding site of LAT3. So here I show you this C225A and a W252 mutation. This mutation still could complement the LAT3 CIDS invisibility in the LAT3 mutant. So this indicated that this E2 binding site are not important for its function. Then this LAT3 may not function as a classical E3 ligase. It may compete with another E3 ligase that binding to the CRCK3 for ubiquitination and degradation. So indeed, through proteomics, we find PUB44, which is a close homolog of LAT3, that's with the PUB5, that interact with CRCK3, and this is the QIP to show their interaction. And also, when we co-express this PUB44 with CRCK3, the protein abundance of CRCK3 was much reduced compared to the control. So this indicated that this PUB44 could destabilize this CRCK3. And we also show that this PUB44 could ubiquitinate the CRCK3, and this ubiquitination most likely is a monoubiquitination, as only one band was detected, and also occurs with UB2 K0 mutant, which no lysine for the polyubiquitination. And more importantly, we found that this LAT3 could suppress PUB44 mediated CRCK3 ubiquitination. So this explains that the function of LAT3 to stabilize CRCK3 is through suppression of PUB44 mediated CRCK3 ubiquitination and degradation. And then we also try to understand how this LAT3 regulated. When the kinase cascade that could induce LAT3 phosphorylation, and also ROS peptide could activate or deactivate LAT3 could phosphorate or dephosphorate LAT3 phosphorylation. And also we find this LAT3 could form kind of bodies on the plasma memory, and these bodies regulated by upstream kinase cascade. And also these bodies actually most likely the molecular condensate and form the droplet in vitro. And we're trying to figure out the composition of these bodies, and also whether they could activate NIRs. And then again, what's the physiological function of CRCK3 and LAT3? We show this CRCK3 and LAT3 are important in plant resistance or plant response to Physarum obspori. As you can see here, these two mutants show the high susceptibility to the Physarum obspori infection. Also, this CRCK3 is highly conserved. The silence of CRCK3 in cotton also should reduce the also should enhance the susceptibility to the Physarum obspori infection. So this CRCK3 and LAS3 not only involved in maintain like immune homeostasis through HOMO2 activation, but also important in physiological resistance. So I still have a couple of slides, maybe talk a little about the physiological. Okay, you know a lot about physiological from Jinglong's work, and this physiological oxbory is another species, and it is also a class kingdom fungal pathogens. In fact, the plants, animals, and humans. In fact, a lot of plants and also in humans, there's also physiolosis is the second most common obstetric mode infection. And this Physiodium oxbory is one species, and they also further defined to different former species and based on the host specificity. For example, this Physiodium oxbory breast infection, FOV, is a former species that infect the cotton, and the cotton cause the Physiodium weird of the cotton. Okay, so this recently the outbreak of FOV race 4 is a concern in cotton industry. It was initially identified in California, which may be one of the reasons to lead the job of the acreage of upland cotton in California, and then it spread to the southern state into Texas. Then we try to understand the pandemic of this FOV4 in Texas, and we collaborated with field pathologists and also the cotton breeders, and identified like different isolates in air parcel where FOV4 outbreak happens. We documented their morphology and also test their virulence like in greenhouse. So we did not find like a difference of morphology for different isolates, but they show like bigger variation in terms of virulence. Some of them are super virulent, some are not virulent compared to this classic FOV4 isolates, which was identified in California, and also thanks Jinglong for the GLP, we also labeled this FOV to look at the infection. Seems like 2 hours started to germinate, and then spread to the entire root. And in Texas, we call it FOV4, but we really do not have this any like evidence, at least molecular evidence to name the FOV4. And then we did like PCR, and then use these classic 4 markers for the classification of FOV isolates. Although they were called FOV4, but we find these isolates actually do not cluster together with this classic FOV4, only based on these 4 genetic markers. And then we decided to perform like a whole genome analysis of these Texas isolates with the combination of Immuno and HiSeq and Minia. And to make a long story short, we find that this Texas or this FOV like former species contain same set of core chromosome with other like Physiodium oxbory. And in fact, here this is in FOL, in fact, cotton, and this actually is a human pathogen isolate. So no matter they in fact cotton or plants or humans, they have a set of like core chromosomes, and each in different former species, they also have a set of unique so-called lineage-specific chromosomes, also called supermutant chromosomes. So they are not like important in growth, maybe associated with pathogenicity and host specificity. And of course, these AIS chromosomes are enriched in rich transposon and DNA transposon that has less like gene coding regions. So not surprisingly, this FOV that in fact cotton does not share the same set of AIS chromosomes with FOL that in fact tomato or like the way in fact human. But to our surprise is that these 2 races of FOV, they also do not share same set of core chromosomes. And this AIS chromosome was originally like considered like to evolve independently among different former species and through like a historic horizontal gene transport. But we find like a massive macrocytony of different like sets of AI's chromosomes from different former species, or even like different races of same species, and even within same races, different chromosomes, they also share like high level of the macrosynthesis. So this suggests that this chromosome or this AS chromosome may also have a common ancestor chromosome that drive the evolution of different AS chromosomes. We also did a phylogenetic analysis and find that this FOV, although they all in fact cotton, but they are evolved independently within this physiological experience species complex. You can see here with these two phylogenetic analysis, they do not class together. They intermix with other FOSC, and for example, this FV1 more close with the ones in fact melon and the beans. So this actually raises the question of the host specifics, because with previous thing, AS chromosome is associated with host specifics, but they don't share specificity, and also they do not evolve from the common ancestor in FOSC. And also we find that some of these FOVs could in fact of different plant species, and FOV1 seem like most variant to a broader plant hosts. And also this may explain why physiological spory may be a multi-host pathogen. Some pathogens in fact plants also could cause infection in animals. So this may be because they also have although they have different AIS chromosomes, but the AIS chromosome may be evolved from a common ancestor. They keep different factors could in fact different hosts. Okay, that's all research I'm going to talk about, and also I would like to thank the people who did the hard work, and also collaborators. And this is my current lab members, and also the funding agency. Okay, and also thanks for your attention, and welcome any questions you may have and suggestions. Yeah.
We have time for questions.
I'm sort of curious about the long range effects of sensing PAMPs or even effectors. It looked like a lot of the markers, cell death markers, or even peroxide was sort of had local variability. But then when you showed the phenotypes of the plants, they're all dwarfed, but they look pretty symmetrical. Like growth is uniformly affected in the whole organism. And so how is that true, number one? And then I guess, how do these long distance signals, how are they propagated? And if it's reactive oxygen, is it extracellular reactive oxygen?
Yeah, it's extracellular.
Okay.
Yeah, it's maybe spread to other stable cells. And actually I discussed with Chris maybe different components evolving local and systemic testing signaling or RS signaling. Yeah, and some of them could be important to initiate local cell death. Some of them could be important to spread the cell death. I think what the mutant we find here is it has cell death in both local and systemic. And the suppressor we identified, I think, most likely block both systemic and the local cell death. Yeah. Yeah, I don't know whether answer your question or not, but this is indeed we're trying to figure out where the difference between local and systemic cell death. One thing I can say that this BTI2 we identified is important in systemic acquired resistance. Okay, so that means that for systemic acquired local inoculation could inhibit spread of the pathogens. Yeah, this BTI2 is important in SAR. Yeah, and it might be also important in suppression of the or spreading of the CRS like systemically. Yeah.
So these LS chromosomes, they seem kind of reminiscent of things like B chromosomes in plants. Are they sort of analogous? Are they mostly just a bunch of junk and some things that provide an advantage?
No, definitely not junk, because I believe people did experiment, if you remove it, is no pathogenicity. Yeah,
Excuse me?
if you remove it, Pseudonym of spori lost the pathogenicity.
Right, but they
Yeah.
don't die, right?
They will not die, they will not die,
Right.
they will not die. They still survive, yeah.
Right, I guess I'm wondering to what extent these might be kind of selfish elements that can drive them. So B chromosomes can drive themselves.
Yeah.
Independent of the advantage they might provide.
Yeah. That's a good point, yeah.
Which is to say, you might find them in situations where what are they doing? They're not doing any good right now. Maybe they don't have to
They do any good for the packaging or for the plants.
for themselves. They
For
do
themselves,
well.
but they are doing good because that's important for pathogenicity, yeah.
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 They get food, right? Cause
Yeah,
disease.
they cause disease, yeah, they got important, like if you do this chromosome, the pathogen will not cannot infect the plant, yeah,
Mhm.
yeah.
Dr. Chen?
Gee, I have a question about you, what is the name of the calcium channel,
CNGC,
So in your model, you're showing that EPL2
Yeah,
can
phosphorate,
phosphorylate and then activate.
yeah.
But at the very beginning, you also showed the backbone also can phosphorylate and then suppress its
Yeah,
activity.
this backbone phosphorus actually degrade and promote degradation. Rolling level is reduced.
Also
Like,
the phosphorus.
also slow phosphorylation, yeah.
So is it because of just like a PKL1, you have different phosphorylation side?
Let's give the phosphorylation sites, yes,
And then you
yeah.
have
Yeah, even
total
I think
opposite effect.
one is the intermiddle, one is the intermiddle. Not, yeah, different, yeah. Because the thing is in Y type, this backbone have to keep all these components level low, otherwise it already cause their death, yeah. Yeah.
So from last part story, so-called those resin in the Fusarium species complex, they have the same set of effectors to target conserved proteins among plants or animals, or do they have a different set of effectors that can differentiate different targets between plants and animals?
Yeah, that's a good question, but I don't think we have a clear answer for that. So, the people think these AS chromosomes actually associated with pathogenicity and a lot of effectors. And when the original Nature paper reported, they said that this actually AS chromosomes enriched with the effectors. But when we look at it carefully, it's not really enriched like effectors. Yeah, and also I don't think we find a clear effectors associated with plant passaging or effectors associated with human passaging. Yeah. We did not see very clear, at least we did not identify, this might not be also be one or two effectors. This is very complicated, it seems like, yeah.
All right, with that, I think we conclude. Let's count in again.